All right, everyone, this is the last session we're heading into. I'm Al George, as you've heard earlier, and I've taken over the reins as Master of Ceremonies from Dr. Bender, who did a wonderful job. Would we all agree with that? I have the pleasure of introducing a series of amazing speakers in this session. I suspect a lot of the families attended this year's FamilieSCN2A conference to learn about what are the new opportunities for treatment. Do I have that right? Interested in treatment? We are too. As scientists, we've been absolutely amazed at the explosion of new strategies at the molecular and genetic level for treating genetic disorders, possibly including SCN2A. And you're going to hear this afternoon some mind-blowing results from scientists and others who will really give you great hope of the future. We're going to lead off with Brian Gardiner, who is father of a child with a rare genetic epilepsy, Dravet syndrome, who is going to share his story about their decision to enroll their child in a clinical trial and what went into that. So give a round of applause to Brian.
Thank you. I appreciate the opportunity to speak to you, and I hope that a little bit of our story, you can find a little bit of your story in it, because those who have children with rare disorders are a unique group. Our daughter first started having seizures when she was 7 months old, and they started infrequently, and then they became monthly, and then they became weekly. And by the time she was a toddler, she was seizing tonic-clonic every day. And we got more and more frustrated as we tried one anti-epileptic drug after another, and we're not seeing any good control until one day when she was 2.5, we were actually in the hospital for a month with status issues and conditions that a number of you have struggled with as well. We spent the better part of 7 years working with our local medical professionals to try to make sense, given the technology that was available in that day and age. We even went to outside experts in the largest metropolitan areas trying to find some guidance as to what was the underlying cause and what were the opportunities. And those who know the history of Dravet syndrome will recognize that there were places and pockets that could have identified this a little bit earlier, but it really wasn't until 2012 when a resident from Pittsburgh walked into our local neurologist's office for her residency and said, "Has this child ever been tested for SCN2A mutation? Because she looks like a Dravet child." And that really opened up a watershed, because it allowed us with a diagnosis to reconsider not only what was going on, but what we might do in the future. And that included looking at the possibility of experimental treatments, which can be very scary for some, because there's a lot of uncertainty with what we have, but there seems to be even more uncertainty with that which we're just beginning to learn. But we now had the opportunity to fill in some information into clinicaltrials.gov and find out what was going on in the medical community. So our story of enrollment is one that is pretty straightforward. There's 4 phases that a treatment or a drug goes through. There is a progression towards more people becoming involved and more information on the safety and the dosing and the potential outcomes becoming available. And every family has to weigh the risks and the benefits, not only in their own hearts, but in consultation with the experts that they're working with. So we made contact with the Nationwide Children's Hospital here in Columbus, and that's how we started. We made inquiries. Everyone's very busy. They're tremendously busy there. We ended up in the emergency room about a month later. And one of the nurses said, "You know, we've got a trial going on here for Dravet patients." And we said, "Yes, we do. We've been calling pretty regularly trying to get enrolled, and we did get enrolled. And thank God we were one of the last ones in the phase 3 trial. And I say that because even though we knew that it was a double-blind trial and we knew there were no guarantees, 1 month after we enrolled, she stopped seizing every other day. So what's a trial really like? Well, you end up collecting a lot of data. There were weekly seizure logs that we had to phone in. There were biweekly visits, but then as the trial progressed, those visits and that extra supervision started to become more and more infrequent as the researchers became more confident of the results, and the need for that safety net became less and less. So one of the things I want to communicate is that the time that's spent is significant, but there is a safety net there. And so that uncertainty that you might have about the experimental, know that there are a lot of good people who are watching out for you. Most trials will last a year, as it was because of a variety of reasons. The trial continued for a full 4 years before there was a review. And the one thing I want to point out is that as you go through that, if you are getting positive results, you need to be thinking about what comes next and asking, how are we going to stay on this medication or have access to this treatment? And your care providers will help guide you towards making the necessary arrangements to work with Medicaid and or your insurance in a way that will work, because our society does care for those who need. We were blessed to be able to actually watch the scientific advisory group look over the results, summarize them. And one of them actually said, this is a slam dunk, no brainer. I vote yes for approval. That made us feel really good because we knew that it made a big difference for our daughter. So for us, the outcome was very positive. Anna's seizures, as I mentioned, were reduced by about 45%. But more importantly, we had an improvement in the quality of life. She was starting to sleep through the night, and her seizures were not happening during the day. So the risk of falls was greatly reduced, and our stress level came down. But it could have been different. These are the published results of the research trial. And I just want to point out that we're very middle class. The median response was a 51% reduction in seizures. But in every trial, there are some that do better and some that do worse. And I'm pleased to know that there are some that benefit from each of the treatments that get tried. But I also want you to know that if you get in a trial and things go in the other direction, there's a safety net there. The doctors, the caregivers, they're monitoring at a high degree, and you will be monitoring your child in a high degree. And really the downside risk is pretty minimal, because if there are negative responses, they pull the child from the trial. So what does it mean to succeed in a clinical trial? A lot of you may have, if you've had children that took a while for diagnosis, may have seen that there isn't always an agreement between the way the population responds to an individual drug, either positively or negatively, and the way your child does. Every patient is different. You know your child better than anyone else. But by talking with your providers, by communicating, you start to understand that research doesn't guarantee a miraculous result, an all or nothing cure. But what it does do is provides greater clarity on something that may seem very uncertain right now. And what I want to leave you with is the sense that science does give us very good guideposts, and we are making continual progress. In fact, one of the talks earlier today, the SCN1A database that was talked about earlier this morning, I remember in the early days when that was being set up, I saw it 5 years ago, and my daughter's mutation, which happens to be a rare missense, wasn't in there. It's in there now. And that means that if not now, in a few years, there will be clinical guidance that doctors will be able to provide for people like my daughter. And that's a wonderful thing. So we're looking towards the next trial, because we're not there yet. You know, seizing 20 times a month sounds really good compared to 5 years ago, but it's not the end of the journey for us. So we define long-term goals, but we have new ones, because with each improvement, there's more hope. there's more opportunity and there's more growth. And that's what you can expect. So consult with your caregivers, consult with the experts, find out all that you can. Look inside your hearts and pray about which way to go, and it'll become clear to you, I think. So I hope you all go boldly on your journey. I know that there was no social connections in my daughter 5 years ago before she went on CBD. And she now has a best buddy, a BFF, and they get together and they play baseball. So there are great things that can happen. So if you have the opportunity to do a trial and it's right for you, by all means, move forward. Thank you.
Yes, thank you very much. Welcome to Columbus. I was so excited when Leah told me that the meeting this year will be in Columbus. And, I moved from Switzerland here 11 years ago, away from the mountains and the lakes to the cornfields. And my colleagues were asking me, what the heck are you going to do in Columbus, Ohio? Why can't you go somewhere cool? And I hope that's what they said. I hope that you saw in the last couple of days that Columbus is actually, it's a quite amazing city. It's growing very fast, but also Nationwide Children's Hospital is an amazing place to work. And I'm very excited that you got to enjoy it here as well. Next slide, please. Or this is working, okay. Let me see. Okay, there we go. All right, that's the hospital. And the title of my talk is CuATSM for SCN2A. However, very similar to Kevin, I'm going to talk about different things. I'm going to talk a little bit about the in vitro modeling systems that we're using and what happens to the patient cells and the skin biopsy that we're getting from you all and why they are important. And then also a little bit just on the gene therapy landscape and timelines and how that has changed in the last couple of years because of the experience that we gained. So very quickly, when we scientists talk about in vitro modeling, what does that mean? This is just a simple Google definition. Basically, we're just looking at things in a dish or in a test tube. So not in a mouse, not in a rat, no fancy hats, just putting the cells into a dish. And ultimately, we were very happy that we were able to also participate in this meeting with collecting 14 skin punches, and there is more to come. Hopefully, they can also be shipped to us. So what happens to those punches? First thing is to get a very, very small area. Let me see if I can go with the laser here from the skin. I was told it doesn't hurt very much. Happy to try myself. And then the punch goes in one of those test tubes, which you might have seen if you went to Nationwide. And that's what when it comes to my lab. Ultimately, we're taking it, putting it in a dish, cutting it up in smaller pieces, putting it in a smaller dish, and then we wait. And it's, I'm a scientist and scientists are nerds. It's still always exciting for me to see how the cells basically crawl out of the skin punches. So you can see here, those very dark areas. These are pictures that we took with our cell phones through the microscope. And so where it's really dark is where the skin punch is thick and the light cannot go through. And then you see those little thingies here on the side. Those are the cells that are crawling out into the culture medium, into the soup, to have better access to the media. And this is a fully confluent dish of cells. Keratinocytes are usually the first ones to come out, and then the fibroblasts follow. And this is, again, a little bit of a nerdy thing. I'm doing science biology since over 20 years now, and I never get sick of looking at the cells in a tissue culture dish. This is a cell that is about to divide, and you can actually see the chromosomes, so the genetic material that is pulled into the two new forming cells. So once we have those cultures and they grow oftentimes pretty well, we can freeze them in large amounts in multiple vials, and we can share them with each other. And unfortunately, oftentimes the sharing takes a bit of time because the universities have to contract with each other, and that's sometimes a little bit complicated, but the goal is to really share all these cells with each other. And we can use them for studying disease mechanisms or for therapeutic testing. Why skin cells? Why aren't we using other cells? Now, it is possible to use other cell types, but skin cells have been used very successfully. It's relatively non-invasive. They're easy to maintain and ship between different labs, and they also recover well from the freezing process. So those are advantages over some of the other cells that are sometimes used. Also, what I like is that they share actually a common lineage with the neurons during embryogenesis. And then there are many available protocols on how to manipulate these cells and actually make cells of the nervous system, because it is a commonly used cell type. So why are these cells so important? And why can't we use just one cell for a specific mutation and want to have multiple cells and hopefully be able to compare them? So what we're trying to do is to understand patient subpopulations, so that we can potentially predict what is the best way to treat the patient. This can be specific to a certain drug, but it can also be non-specific, because it's not just the drug that matters. It can also be that certain patients have less uptake of the drug and things like that, that we could also test, that have nothing to do with the mechanism of the drug itself. And the other thing that people sometimes forget is the genetics reports give us a lot of information, but they cannot always accurately predict what actually happens in the cells on a phenotypic level. So we want to understand the impact of a given mutation on the disease process per se. And sometimes we stumble upon things that are very unexpected, and those can be leveraged to develop new innovative therapies. I'm going to give you one example for Duchenne muscular dystrophy. It's a muscle disease that is caused by a protein that is also very big, so you can't put the entire protein into a gene therapy because it doesn't fit. And this story is based on the observation in a patient that has a very early stop codon in the gene. So technically, this patient shouldn't make any protein and should have a very severe disease phenotype. However, this patient was discovered ambulatory, I think in his 70s, so very, very uncommon. And based on the mechanism that the researchers at Nationwide found, they were able to develop a gene therapy. And they have recently published these results at ASGCT. And based on the gene therapy that they came up with, they have now patients that are able to produce full-length dystrophin protein with this gene therapy, which is really very exciting. This is another example I want to give from a disease that we're working on, which is called NEDAMS. It is a disease that has only been recently discovered. And I don't want to belabor you too much about the protein, but ultimately it was supposed to be a haploinsufficiency, meaning if you have a mutation in one of the genes, the other gene will not produce enough protein for the patients to be healthy. When we got the cells from multiple patients compared to healthy controls, we didn't see a reduction in the protein levels, which was strange. We're like, this is supposed to be 50% of the protein levels, but it is not, except in one patient. And to make a long story short, what we found is that instead of having less protein, the protein is in the wrong place. It's actually, instead of being in the nucleus where the genes are and changing gene expression, it is now being sequestered into other areas of the cells. And when we overexpress the patient mutations in a healthy mouse, okay, here we're using a mouse, but this is a healthy mouse to start with. When we overexpress the mutation, we actually did get the disease phenotype. And why is that important for us to know? I think it's really important for the gene therapies, even if it is a supposed loss of function mutation, the remaining mutated protein can actually impact the efficacy of your gene therapy. Sometimes you will have to reduce the levels of the mutated protein or RNA in order to have the best effect, even if you're trying to increase the expression of your gene. So what we're trying to do with these kind of methods is to help clinical trial design. That's our future goal. We want to really implement the disease modeling into the clinical trial design. And also, of course, you have seen there's lack of animal models. We can't have a mouse for every single mutation. We also need to look at the different cell types that are potentially involved in the disease. And then one of the things that we're trying to do in my lab as well is compare the cellular phenotype, not the genetics, between different diseases and see whether we can leverage experience and drugs that are in clinical trial in other diseases for SCN2A and others. So we're using a direct reprogramming method where we take the skin cells and push them into neuronal progenitor cells. We're not making induced pluripotent stem cells. Not going to go into the details of the differences. Happy to talk about it if you're interested. But this is a system that has been really well established and validated for testing in several labs. We have multiple types of methods that we can use. We make astrocytes, we make neurons, we have different ion channel uptake assays, we have metabolic assays, and we have assays where we look at the interaction between different cell types of the nervous system. Okay. I talked about this in previous meetings as well. So this is actually an old graph. I'm not going to show you a lot of things, but by doing these comparisons between different diseases, we found a drug that is already in clinical trials for another adult onset disease that seemed to be helpful. The symptoms that we found in the SCN2A patient cells that we were interested in is increased mitochondrial activity, so ATP and basal respiration. And the drug was able to reduce that energy problem. And that's the same thing that we've seen in another disease where this drug is in trials. I'm not going to talk about this, but we are currently working on evaluating the safety of this drug in juvenile large animals because the drug is currently in clinical trials for adult onset diseases, and we wanted to make sure that it will be safe, hopefully also in younger patients. And that's what's currently happening. These are the signature of our little piggies that we have to use. Again, we want to make sure that the children will be safe if we're moving forward with this treatment. What are some of the other focuses in my lab? We're also testing additional small molecules. It is not a drug screen. It is theory-based, non-random, as I said, based on observations. We're always looking for new industry collaborators that are interested in this system. We have cells from a large pool of epileptic, it's autism and seizure disorders that we can compare at the same time, which I think is attractive for collaborators. And we're trying to categorize these diseases and subtypes into different groups so that we could move faster in terms of predicting potential new applications. We're also continuing the development of gene therapies because I am in the Center for Gene Therapy. And I'm not trying to reinvent the wheel, of course, and that's something that is really beautiful here. So I'm talking after Kevin, before Chris. We're trying to really stay somewhere in the middle and do something different that is not overlapping with what they're doing so that you can increase the probability of success. And then one of the things that we're also focused is to try and find assays that are as short as possible so that we could more easily implement them into clinical trials. This is an example from a different disease where we had an assay that was able to predict an outcome in the dish for a pathogenic variant. And then we found a patient that had a variant of unknown significance. And I know Kevin Bender is probably going to have a heart attack if I call these things neurons, but we established an assay that allows us to push the fibroblasts into neuron-like cells or induced neurons within 7 days. Of course, they are very rudimentary. Maybe you can compare it to my 2005 Ford Explorer versus a Porsche from 2020. They both drive, but of course it's not the same thing. However, using this assay, we were able to establish that the phenotype of these cells with the VUS was very similar to the known mutations, and that the response to the treatment was actually there in addition. Hopefully these kind of assays could be used again because they're much shorter in the context of clinical trials. Getting to the last part, talking about the gene therapy and the timelines around gene therapy, maybe a quick reminder, what we're doing with gene therapy is we're using most often for nervous system a virus, and we're removing any of the genetic information from the virus to replace it with a therapeutic gene or a therapeutic RNA that can act on the promoter or other things like that. The surface is basically viral, but the inside is what we want to deliver. And we're using the virus as a mailman, basically. There are other transport vehicles that are not viral, but to this point, I would say for the nervous system, they haven't been as efficient compared to the viral delivery systems yet. When you look at the timeline of a just research project, usually it all starts with an idea. We need to get the funding for it. We do a small proof of concept study, then we might need to get more funding for it. We do a larger proof of concept study, we might get more funding, and then we go into the safety studies. And every step de-risks a little bit the project as we move forward, but it takes a long time. And the problem is also at every step, something can go wrong. It can look really promising in the beginning, but then in the larger study, it can turn out that for some reason, with a larger number of animals or a larger number of cells, you won't see the same thing. Or it is really promising, but then you have a safety problem, which means you have to go and redesign your approach, and then you start from scratch. Well, not exactly from scratch, but you have to go back. And after you do all of this, oftentimes, or at least steps 1 to 4, that's when you start trying to get funding for your clinical trial. And that's when you start worrying about manufacturing for the clinical trial, which per se, again, takes about 2 to 3 years. So you can see the total timeline, if there are no hiccups and no COVID, you end up between 4 to 6 years. There are ways to shorten this timeline, which are not increasing the risks of the patients in the clinical trial, but they are bearing an increased financial risk, which is if you start your clinical manufacturing at a much earlier time point. If you're comfortable with the delivery system and you know where it's going, you can actually, and you're very convinced that your strategy works well, you can start clinical manufacturing right at the beginning, right? Now, the problem is if you have any safety or efficacy problems, you will have wasted all that money. But you can very significantly shorten the timelines when you do that, and that's what they did for the COVID vaccine development. So when you do this without hiccups, you look at about 1.5 to 2 years. The problem for SCN2A gene therapy, as I see it is a very complex gene therapy because the size of the protein, unfortunately, is big, but it also has a very complex function. So we can't just cut pieces out that make it smaller. That's very, very complicated. And then again, we have those different effects, loss and gain of function mutations. So there's not going to be a one-fits-all solution. So we probably need to think of multiple solutions for the different types of mutations and subpopulations of patients. The other thing I will say is that even though gene therapies has been in the news a lot in the last couple of years, it is still a very young field. So a lot of the trials, when you look at it, they are still in early stages. So phase 1 safety or phase 1/2, which is safety and exploratory outcome measures to see if it's working. And we don't understand everything about these therapies yet, and there are safety risks at these early stages. And this will continue for a little bit more, so we're still, again, early stage. We're not going to have as many outcomes in the next couple of years. There also, in addition to that, there have been severe side effects in clinical trials, in intravenously dosed trials with very high vector concentrations. And those have been, up to fatal. So it is something that is very concerning. Again, it has happened at very high doses, not at the lower doses, but it's definitely something that has increased the bar of the safety studies. So we need to do more than what we had to do 3 years ago. It has increased also the bar of the testing of the clinical material. We need to be sure that we can actually properly analyze the clinical material, know what's in there. All of these are good things, but they do add to the timeline. And it's also become more complicated to find financing partners because of the risks. As Kevin said, the delivery methods are absolutely key. There are a lot of companies that are working on this and a lot of researchers that are working on this in academic institutions. For neurological diseases, there are more direct delivery methods, so you don't have to go intravenously, which I think will definitely be beneficial for safety concerns at that point. We're also here at Nationwide very interested in novel delivery methods. I will say because of those new standards as well, the manufacturing time and the release testing of the clinical material has become longer as well. So the FDA, because this is a young field, is learning with every program that they see, and they're adjusting the requirements every time. So even if I give you an estimate today, this is going to take this long. If 6 months from now, the FDA changes what they want to see in terms of analysis, that can add another half a year right there. So I know it's frustrating. We can't give you exact timelines, but I would say at this point, clinical manufacturing and testing until we can get into the clinic is about 1.6 to 2 years. The good part is though, as I said, FDA is learning, and that also means this could shorten again. Once they get more comfortable with more programs that they're seeing, they might remove some of the requirements again. So this is not a permanent state, but it makes things a little bit complicated for gene therapy at the moment. Yeah, that's all I wanted to say. Again, very excited that you're all here. Thank you so much for Families of SCN2A for the support. Thank you everyone for participating in the research. It's really important. And yeah, this is my beautiful lab. We just had an outing recently here in the area. Thank you.
Great. Thank you, Dr. Meyer. Our next speaker, Dr. Chris Ahern from University of Iowa, is going to present some new ideas about genetic and molecular therapy of SCN2A. And we're pivoting away from DNA as the therapeutic molecule to RNA as the therapeutic molecule. So stay tuned. Buckle up.
Yeah, buckle up. So first of all, thank everyone for having us here and allowing us to come into your amazing meeting. It's always so inspiring to get a chance to mingle with you. And we take a lot of this excitement back to the lab. So it's just really wonderful to be here. I'd also like to thank the dads from last night for allowing the scientists to crash your party and just to see what a great community you guys have put together here. Especially the Chick-fil-A at the end of that was a very nice touch. Very nice job, Will. To start things off, I want to go into this general idea of what we've been working on is kind of agnostic to the disease. There it goes. There it is. And so what we've been working on is an approach to repair stop codons. So we've heard a lot about different types of molecular lesions that we find in SCN2A. Loss of function mutations, in particular stop codons, are what I'm going to be talking about today. And this is really a collaboration between myself and Dr. Ashley Williams, who's in the audience at the University of Iowa. So we've been really excited to collaborate to bring together clinical and basic research together. But when we look at, I'll just let this go out, premature termination codons in rare disease. So when we look at the rare disease space, we're talking about thousands of diseases like SCN2A syndrome. And within those genetic diseases, stop codons, or what we call premature termination codons, represent 10 to 40% of those populations. So if you look at all the loss of function mutations, you see that stop codons actually show up a fair amount of time. And when you look in composites, so we think of rare diseases, but as a cause of human disease, stop codons we think could affect upwards of millions of people on the planet. So each one of them is rare, but the cause is shared in a way. So you're within this greater community of people suffering from a very similar kind of molecular lesion. So what I want to talk about today is kind of an approach that we're really early on, but we're developing it, that could be used eventually to correct not just SCN2A, but all types of stop codons. And PTCs, and and we're talking about neurological disorders, 400 gene stop codons will cause different types of diseases. And so when we look at sodium channels, I can also thank the speakers that came before me, Dr. George, of course, and many others who also articulated really nicely, the topology of sodium channels. We work on SCN2A a lot in the lab and SCN1A, but you can see here just highlighted all the different positions of where stop codons are known to show up. They just really are a buckshot of insults against this gene, which makes it especially difficult to treat because each one of these might otherwise demand its own type of treatment. And so that kind of highlights the importance of finding something more fundamental and disease modifying in order to kind of take care of these disease mutations. And so what this plot shows is basically premature termination codons in just all of epilepsy and highlighting a handful of genes here. Each one of these is a different gene, CACNA1A all the way down to SYNGAP. And I'm going to draw your attention right here to SCN2A. And what I'm showing you here is, I'm sorry for the small print. What you can see here is in this first category is a percentage of stop codons in that disease. So for instance, something like CHD2, 40% of all that genetic disease is caused by stops. In SCN2A, this number is highly variable. It ranges between 10 and 20% of the genetic component are stop codons. And that really depends on the database that we look at. So that kind of highlights the issue in epilepsies, that you have this high variability, but it's pretty significant within the population of genetic epilepsies. When we look at the cause or the type of stop codon that you can get, what you can see is all along here, these are all the different types of stop codons that can occur in the human disease space. And the most common is arginine TGA. So that will generally represent 40% of all the stop codons that are with any given cohort of stop codons. So arginine TGA is a real big target to go after, because if you could find a way to systematically repair an arginine TGA, you would have an approach that would be generally applicable for not just SCN2A, but really many, many diseases. So ARG TGA is really at the top of our list, but then there's a few that really round it out. So you can imagine in this list, maybe there's 5 types of stops that represent over 80% of all the known stops. And then they become really quite rare. And we can talk about this later. This kind of clustering up here is really for biochemical reasons. And so the challenge of like, when we think of CRISPR or normal types of gene therapy, first of all, SCN2A is too big. You wouldn't want to just throw it in a vector and overexpress it, because it has to be very precisely regulated. But if we look at, if each one of these black lines we consider is one of these different genes, all of them would have different positions. So if this is one patient, this is another patient, another patient, another patient. There's this spread of different types of contexts, and CRISPR requires a context to work. And so that's the challenge with gene editing. It's going to be really hard to go after all these rare mutations with gene editing. Small molecules have been out there and used for a while. One's called adalurin. They promote just kind of a messy read-through of stop codons. Those haven't been as successful in the clinic, and they also put the wrong amino acid in. So they basically bargain a missense for a nonsense. And we all know in this room that a missense mutation in the sodium channel is not a great outcome either. And so adalurin and those read-through drugs are really kind of off the table for now. And so what we wanted to come up with a way is if we could use transfer RNAs. So we've all heard about RNAs. Many people in this room have probably been injected with RNA. We just did a big experiment on the Earth where we injected over 1 billion people with RNA as a massive clinical trial, and it seemed to work out okay. And so tRNAs are just a small RNA. They're around 75 bases. We call them tRNAs because their fold looks like a classical T shape. And so what we wanted to try and do is to understand if we could use them to fix these stops. What tRNAs do for a living in cells is they work in this is like a rutabaga shaped thing. This is called the ribosome. And what the ribosome does is it turns RNA into protein. And how it does that is through tRNAs come in here and they read this language, and then they turn that language of DNA into the sequence of protein. And if you have a stop codon in the middle of your RNA, you have a truncated protein. If we do that experiment with a fluorescent protein called GFP, green fluorescent protein, we put a stop codon there, it goes black because we've knocked out its function. But what we wanted to do is, could we edit tRNAs systematically so they replaced this stop or this pause with the correct amino acid, allowing it to basically generate the wild type protein. And so that's what we've done. We've generated a platform, and I'll talk about that next, essentially where we have arginine tRNAs that no longer read their cognate kind of sentence that they're supposed to go after. They now convert in-frame stops back into their reading frame of what they're supposed to be. And we did that by basically systematically looking at the human genome, looking at the 500 or so tRNA genes that are out there, and then scanning all of them for their tolerance to decode stop codons. And that's what you see here. So this is my messy slide apology, but this is the platform that we now have to work with. And what we have here is by color, these are different types of tRNA families that are here. And you can see here, there's arginine is this family right here. So there's almost 30 arginine tRNAs in the human genome. And we found that about 20 of them work really well as UGA suppressors. So what that means is that within this cohort here, you have tRNAs that convert that disease stop back to arginine. And it works at the level of RNA. So you're not changing the levels of the protein. It's working at the levels that are already regulated in the cell. We also have a great cohort of tyrosine in 2 different flavors of stop codons, as well as coverage of every known human stop codon that can cause disease. There is a tRNA to repair it and to repair it back to its wild type amino acid. So that's the promise, that's the excitement. So now a lot of the hard work is delivery, testing, fidelity, all those other questions. So just to get at a very simple experiment, how well does it work in simple cells? So we picked 2 sites that are arginine and SCN2A. So here's wild type SCN2A. We all know from Al George's lecture what all of this means. So I'll quiz you on this later in the inward and an activation phase of the bar session. And so this is a wild type. This is what normal channels look like. If you put a stop codon in there, there's no expression, none of those squiggly lines. But if we just express either of these channels with this disease stop codon with one of our ARG tRNAs, just kind of throw it in there with it, what you see the next day is basically complete rescue of that RNA. So now that no longer is that stop valid, but that stop has been read through, and it makes the wild type protein in its place. And you can see that kind of wild type behavior there. So then the big question from this point out is, how do we develop this? How do we move forward in this community to make this something that's valuable for general use? And so the first thing we've done is we've worked with the community to develop 2 new mouse models. So we have tyrosine 84X, thanks to the Parks for their inspiration. So we've made this new mouse model that we're using now to test drive these tRNAs. This mutation is associated with epilepsy and developmental delay, ASD, intellectual delay, and sleep disorders. And so, this is a really important mutation for us to study. It's right at the N-terminus of the channel. And we have a second one to balance it again. So we're basically looking at 2 different stop codons. We developed those mice. When you look at those, they are haploinsufficiencies. So for instance, the ARG1626 mouse, if you look at SCN2A levels, these smudges are Western blots that show how much channel is there. You can see that you have less channel when you have the ARG1626. And then we've also developing different cell culture systems, IPS lines, which I think are very important because as Dr. Bender told us, we're not here to cure mice, we're here to cure people. And so we look to IPS lines. We have a number of neuron cultures that we can express and nucleoaffect different tRNAs into them. And so we've become competent at doing that. The other thing we can do in mice at the same time is behavior, right? So we can do all sorts of behavioral analyses. We're starting to see differences shake out in the 1626 line in different ways and shapes and form. Mice are not as smart as rats, so we can't train them to do as many things, but still there's behavioral markers that we can look at correcting. And also we can look at mouse behavior with pup vocalizations. So baby mice will kind of talk to their mothers, and it's been shown with ASD that there would be differences in that type of language that they use. So John Sikora, a med student that we've been working with, has developed an assay that we can look at different spectrograms of these ultrasonic vocalizations of how they speak to their mothers in the cage. And that's another way that we can look at correction. And then lastly, there's 2 slides that we've been looking at IPS lines. So we have cells, tyrosine 84, as well as the C959 line that his lab is using. We've developed these as well. So we have these platform systems now that are really in place to start testing the hypothesis of how would we go about, packaging, delivering, and expressing tRNAs, looking at on-target and off-target effects to see what is the next step going to take in order for us to build this out into a really viable therapy. And so with that, I'd just like to thank everybody, obviously the Williams lab, the folks in there that have been doing the IPS lines, and John for doing the mouse work, my lab, of course, for all the work they've done on tRNAs and sodium channel biology. It's been amazing. And then also collaborations. It's a key part of the whole SCN2A community. We've been working with a number of folks at both at University of Iowa in our behavior core, but also in other places. Catherine Meyer, of course, you just heard from University of Iowa, lots of sources, as well as the FamilieSCN2A. And thank you for your attention.
Thank you, Chris. Our last scientific talk in this session will be Dr. Yang Yang from Purdue University. He's going to update us on his work in his laboratory on modeling SCN2A disorders with mouse models. And one of the things I hope he gets to is using those models to explore the reversibility of the phenotype. Dr. Let him go, Leah. Dr. Yang.
So I heard that the IT have some issues. I have to talk about some random stuff for a couple of minutes before we go on to the real scientific talk. So I think Dr. Banner sings better than me. So if anyway, so it's my incredible honor to be here. It's my honor, and actually it's a privilege for me to stand here to address to the patients. It's really, I receive a lot of support since I started my lab in 2017. It's a lot of support from the foundation and give us fundings and let us know the patients give us this responsibility that we should working for the cure. The IT is still not up. I'm running out of words to say. So for today's talk, I'm going to discuss my research both using the mouse model and iPSC model to address some of the clinical issues. We are actually really laser focused on finding therapy, figure out disease mechanism, identify new targets. That's what we're going to cover in today's talk. And I also want to introduce that I have many students. I think more than 7 is coming this time. So they have posters here after you hear my talk. If there are certain things you are interested in, you actually can go talk to them from the poster. Actually, the slide is up. Great. Again, I want to thank the SCN2A Foundation for giving me this opportunity and all the support. Actually, the title is the direct work from Leah. You know, that's a beautiful title. What's the cell and animal model of cystic tolerance, right? I used to have a scientific title, which quite boring. And this title really gave me the opportunity to focus on the real important issues that we're going to discuss and that is matter for the family. Okay, so many of you guys have been heard about the ASD. Actually, it's a very prevalent disease. It's affects 1 in 44 people in the United States. It's affect a lot of people, and in many of these autism, people have impaired social interaction. They have impaired communication, and many times they have this repetitive behavior. So that's a definition for ASD. And you guys may already notice in your children, right? There's a large portion of the kids with this autism, they have epilepsy. And this is a major comorbidity that making the life really difficult. And there's a studies have found if we can really just treat the seizure, we can significantly increase the quality of life. So that is understanding how the seizure occurs and how we control them. It's a major goal in the laboratory. So as you may see, some of you guys see this is a very important and the popular slides, many scientific talk they're going to show here, which shows the different type of gene has been mutated in the autism cohort. You can see from these slides, there's a group sequenced more than 35,000 individuals. It's a large number. And they find a lot of genes had been mutated. If you look at the x-axis, which is the protein truncating mutation, just like Chris just mentioned, you have stop codon, you have the fringe shift, that's protein truncating mutation. On the y-axis, that's the missense mutation. Dr. L. George has mentioned that that's a lot of them. And you will see SCN2A it's very high on both the protein truncating as well as the missense, making it almost the #1 leading gene involved in all these autism cases. So that emphasizes the significance of SCN2A in the whole autism community. So you guys also see that a very nice plot is made by Dr. Bender. Actually, that is the current dogma suggesting how we view the SCN2A mutation. On this yellow side, you will see this gain of function. They cause neuronal hyperexcitability, meaning your neurons fire more than they should. So they cause seizure and epilepsy. On the other end, we have this SCN2A loss of function. These kids, they will have intellectual disability, and since it's opposite on this like a gain of function, right? It's a loss of function. So people predict this loss of function will cause a reduced neuronal excitability. So that is the current dogma. But this current dogma do not address one very key issue is among these loss of function kids, about 20 to 30%, they will still develop this very severe seizure. So what's going on? If you dump their excitability, they're supposed to be like a seizure resistant rather than they develop a seizure. So that is a clinical mystery we try to talk on. And I'm going to share some result. Me and Dr. Benner's lab, we figure out maybe the possible reason behind this. So to study, SCN2A, we can study human, but we cannot actually do a lot of experiment on human, right? We need models. So mouse model, although it's not perfect, it's one of the leading model for people to study this disease. And for SCN2A, the model is also we see some limitations. For example, if we cause SCN2A complete knockout, we call it minus mice. These mice, they die 2 days after birth, so we cannot study them. And many labs, they study SCN2A plus minus mice, which is heterozygous. Dr. Brenner has been spent a lot of time discussing. These mice, they have some behavior abnormalities. It's not huge. So to me, I want to study some bigger behavioral phenotypes so that we can really study them and have a bigger dynamic range that we can try to see some rescue effect. So you will see for SCN2A minus, it's 0. For SCN2A plus minus, it's like a 50% reduction. So naturally, you will think if we can achieve a reduction number more than 0, but less than 50, so maybe we will have a mice that are very severely impaired, but they still can survive. So that's what we do in our laboratory. In collaboration with BL scanner, we actually using a new strategy called gene trap knockout. This gene trap will allow us to have a homozygous mice, and you can see from our result, they will achieve about 80% reduction, meaning we have about 20% of this SCN2A will still in the system. So this 20% of SCN2A, they will make the mice survive, but they also make them very abnormal compared to the regular mice. So what we show here is just a few behavioral result here. These mice, they do not do any they do not do any nesting behavior, right? Like an animal, they like to sleep in a very cozy nest. The wild type, you can see if we put a nesting material, they're going to make a very cocoon nest. But for our SCN2A deficient mice, they actually do not even recognize the nesting material. They don't make a nest. They also have other abnormalities, like they have GI issue. Muriel, who take care of these mice in the lab, always report, they have GI issues, that they also have like a sudden unexpected death. And they also have sensory abnormality. I don't have time to show them. So this mice is actually very abnormal, which could be a very good model for us to understand this disease. Also, thanks to Leah, we know these kids, we have sleeping sleeping problems, right? So we actually have a study also figure out in our mouse model, these mice, they actually do not sleep well. And we actually have a study just published together with Leah. So Leah is also on that paper. It shows over there. Then another important thing is, remember, we actually want to understand why this SCN2A deficient will lead to seizure. So that's, I just condensed one complete paper into just a panel to showing in our study, we find in our SCN2A deficient neuron, they actually fire more action potential spikes than the wild type. That is really a counterintuitive finding. When we just figured out, I was, oh, nobody gonna believe this, right? That's too controversial. But we suddenly figure out that Dr. Banner's lab found a similar finding. Then, we each other support each other. Then we actually eventually lead to a study we published together. So this actually result suggesting this loss of function of SCN2A, they actually will cause neuronal hyperexcitability, then cause seizure. And so this is the paradigm you already see. So with our study, both our lab study and the Banner study, we actually kind of shifted the paradigm a bit. If we really cause a very severe SCN2A deficiency, maybe you're going to actually increase the neuronal excitability. That could be the result to this seizure in these kids. And this two paper was published back to back, and was received a lot of highlights from prestigious review journal. Okay, since we are talking about autism, and this is a really important disease domain, we want to see what is the key phenotype of autism, which is the sociability. So here I show this very classical result from the mouse study, that we have something called a 3-chamber sociability test. Mouse is a social animal. If you put a mouse into a middle chamber, you can see here, and then the mouse is going to recognize when mouse is on the one end, there's no mice on the other end. So he naturally want to go to the chamber with the mice to interact with them. And we find for most wild type mice, they have a really high social time. They really want to interact with the different mice. But when we put our SCN2A deficient mice into the chamber, they actually stay in the middle chamber or stay in the chamber without mice. They actually really do not want to interact with any mouse at all. So that's really give us a good hope. These mouse have impaired sociability. They may be, the time goes by really fast. Okay, they may be a really good model. And we actually study different paradigms, receptor interactions, and we do this home cage sociability. All of them show this mouse has impaired sociability. So we want to see like how we're going to do with that, right? Like we know there's a phenotype, how we can rescue it. So in our mouse model, we actually has a very smart strategy to using this genetic modulation to mimic something called gene therapy to restore the expression of SCN2A. In this way, we actually inject the virus into the mice, just like this gene therapy in human, and we're testing for the protein expression level. Indeed, we can see we can rescue the sodium channel 1.2 expression level. And with this rescue, we was able to see in adulthood, the mouse is fully grew up in adulthood. We was able to rescue the social behavior with this gene therapy-like approach. This, I feel like it's a very important achievement, achievement at least in mice, right? Showing in adulthood, we are able to rescue a very important behavior that maybe allow us to move on with this gene therapy for this animal and eventually into humans. So talking about the mechanism why this SCN2A deficiency will lead to these disorders, we found many potassium channel has been downregulated, and we will see this potassium channel expression level can also be regulated if we put the rescue in. So basically we rescue the sodium channel, this potassium channel expression can also be rescued. So we are actually testing some potassium channel open to as a different therapeutic approach. Maybe we're going to test to see if we can achieve any intervention result in mouse. Again, we also use a different way to using this mechanism. We're doing RNA sequencing to figure out what pathway has been altered. Very interestingly, we found that the GABA signal pathway has been altered here. And the GABA is a very important target for many FDA approved drug. So here we're using actually a FDA approved drug called clonazepam. This drug actually was also able to increase the sociability. So if there's any clinician here, you have using this GABA drug to treat the kids that have some positive outcome, maybe you can talk to me. We really interesting to see if the similar drug work in the mice, can they also work in the patient. So this is FDA approved drug to treat the seizure. So I think it could be a really good promising target for the clinician to see if there's an effect in human. And we have a different drug, which is AZD. It's a clinical compound. It's not approved by FDA, but it's targeting the same pathway. And we use this as well to find the positive response here. So we all know like the mouse is not a tiny human with a tail. So we always want to validate and expand our result into human cells. So my student Jeff and Maria, they actually is the driving force to establish this human iPSC model that allow us to test additional drugs. So here we talk about this L1342P mutation. You can see here, this disease is this mutation causes a drug resistant seizures. And in the biophysical property, actually they have both gain of function and the loss of function feature. So it's very difficult to figure out this is a loss of function or gain of function in this biophysical study. So actually have to rely on the iPSCs. So that is just a general protocol, how we differentiate the iPSC into neurons. And Jeff actually tested these neurons in the MEA, which studied the excitability. They indeed find this neuron show a higher excitability and synchronized firing, indicating we really have a good model in the dish that can allow us to test the drug. So what drug we're going to test? First, we want to see if we can recapitulate the clinical phenotype. This patient is drug resistant. So we put this drug called the phenytoin into the dish. And you will see for the wild type neuron, the phenytoin is very nicely inhibited the firing of this neuron. But for our neuron with this disease-causing mutation, the drug is less effective. You're using the same dose. You do not see the similar reduction. So we know something is not helpful. That is good, but we want to see something is helpful. So what we do is we actually find a research compound, which is an NAV 1.2 specific blocker. We call it PTX3. This is not a drug. This is a toxin actually, but this is a 1.2 specific blocker. Using this blocker, we actually was able to see a very strong and robust inhibition of this neuronal firing, which indicate maybe this like a 1.2 specific blocker can be further developed into drug to treat these kids with this gain of function mutation. So this provides some information for the drug company to move further along this line. So then the last slide, I just want to show that we're also moving towards this the bring all organoids model, and you will see in the poster, we develop this organoids model. We're testing new therapy, including the ASO, other things. Hopefully we can move the needle and towards the therapy. So what are the actually SCN2A animal and cell model telling us? First, they're going to review the disease mechanism, allowing us to identify novel unexpected target to treat the kids, including this GABA receptor, potassium channel, and they also allow us to establish this autism and epilepsy in a dish model, allowing us to do the personalized drug discovery, advancing this precision medicine for kids affected by this disorder. So at the end, I want to thank all my lab who performed this study. Without them, I cannot stand in here to talk about this. And they will have their post out there. I want to thank the NIH and the Family Foundation for funding support and all this great support. Thank you guys for listening.
Thank you so much, Dr. Ang. We are now scheduled to hear from the Scientific Advisory Board. I don't know who is the speaker. Sean will talk about, his title is Other Novel Treatments in the Pipeline, N of 1 treatments, and then we'll break for the Q&A panel. So.
Hey, everyone.
Of the SAB to the stage now.
All right, so this session I thought to be just more Q&A from the families. But listening through these sessions, I threw together a few slides because I think maybe there's probably some questions on what is a drug, what is a clinical trial. So I wanted to just go 5 minutes and walk through a few of those things. Play. Sorry. But this session's been fantastic. My first session conference was 2019. and seeing how all these projects have grown is really cool. It's also the first time I've ever heard rutabaga being used in a scientific publication. So Chris, that was fantastic. No, not working. No slides. Okay. All right. I'm all about the interpretive dance. So I guess a few quick points. Clinical trials, what are they? Like, we're getting to the point where drugs are going to start to be developed for our kids, and that's fantastic. That's what we want. So how a drug is developed is they go from a discovery phase, which is really computer-based analytics. And once a drug is designed, it then moves into a preclinical phase. And these are all really standardized terms that, us as families, we have to get used to and understand a little bit as we start to see. There we go. All right. So, really high level how drugs are approved. These are kind of the phases that a drug typically goes through. Start is discovery, it's on a computer, analytics, and then they move to preclinical, which is a lot of what we've seen today. These are in, oh yeah, much louder. So preclinical is a lot of what we've seen today. These are in animals, they're in cell culture lines. And then at some point, it moves on to being a phase 1 trial. A phase 1 trial is usually a safety and pharmacokinetics. So how the drug working in the body, these are often in healthy volunteers, but for rare diseases like our disease can also be in patients. Phase 2 is, after phase 1 is completed, phase 2 starts up. And these are kind of proof of concept. So does the drug work? And phase 2 is kind of where you initially see data that the drug could work in the kids. And then phase 3 is a much bigger trial, and that's kind of to show statistical significance. So they take the data from the phase 2, make it bigger, and get it ready for approval, get it ready for going out in the pharmacy. So once the phase 3 is complete, and if the data looks good, it goes to the FDA, which makes the decision of whether a drug will be approved or not. And then we see it in the pharmacy and we get it for our kids. But phase 1, phase 2, and phase 3 is when kids start to get treated for it, or adults. So different types of drugs, now that we have some perspective on that. So historically, SCN2A, kind of all diseases have been treated with small molecule drugs. That's 99% of all the drugs out there. So these are small chemical compounds that, go through our body, your aspirin, your Tylenol, and they interact with the protein. And, a lot of things have been helped with these. But the past decade, and we saw some of these in some of the talks so far, we're starting to develop new tools that we can now tinker with the process of getting to the protein. So a protein is made from our DNA. DNA then turned into RNA, and RNA then converted into a protein. So the market and biotech is starting to develop these tools that can now tinker with RNA and tinker with DNA and make fundamental changes that can, potentially impact our kids. So these tools are being generated. Some have gotten to the finish line for other diseases, but they're in the process for SCN2A as well. So the developmental pipeline for SCN2A. So these are, a lot of these we've heard of today, but I'll kind of walk through them quickly. Longboard Pharmaceuticals has a small molecule that is currently in phase 1/2. So this is a drug that's in the clinic for adult SCN2A patients, and it's for DEEs. Praxis has 3 drugs preclinical. Prax 562, it's preclinical gearing for phase 2. So it's, could be entering in patients, jumping right to the phase 2 portion for DEEs. Their 222 compound is kind of one of these more new age molecules that are tinkering with the RNA. So this molecule is preclinical gearing for a phase 1/2, and it's for gain of function. A lot of these other ones we've heard in this session today. And then Praxis also has a discovery molecule for loss of function. So kind of the first stage of that trajectory. But I also wanted to do a quick intro, and I think Dr. Olson's going to give a quick snapshot on these too, but that typical pathway that we talked about of discovery, preclinical, phase 1, phase 2, phase 3, there's a little bit of a paradigm shift that's going on for now that we're developing these tools to interact in different ways. The FDA has allowed for N of 1 trials. So we're starting to see some of these. So these new tools are allowing for really precision medicine pretty much for one patient. And Heather can give a quick detailed explanation of that.
Keep this relatively brief. You guys have been very patient, but I think it's an important area to think about. So I'm an epileptologist and focus on epilepsy genetics. And the N of 1 approach is of interest because of these new therapies that are very precise, and some of them will go to the more traditional trials if they're relevant to a large enough portion of the population. And in other situations, there are specific variants or specific approaches that are really only relevant to one patient or a couple of patients. And that's when you get to the N of 1 or N of a few approaches. And it's actually not completely novel in epilepsy genetics, even in the clinical setting. We've done it on a not so much research, but clinical basis, kind of overlap of clinical and research with off-label use of medications and or individualized compassionate use INDs, where we create a very rigorous approach to assessing the patient before the treatment. We treat them, we monitor as we go, and we have sort of outcome measures and go no-go criteria, and it's done clinically. Now, these become more expensive and more invasive, and so needing a really formalized approach, but the goals of an N of 1 are different than a larger trial where you're looking at for FDA approval. In this case, you're trying to treat a specific individual patient. And so it's something relatively new in the research realm. And there's actually something called the N of 1 collaborative that I've joined, and a bunch of people are kind of coming together, led by Tim Yu, Winston Hand, Steve Finkel, a bunch of people coming together to really try in a rigorous way, develop approaches to develop natural history before the treatment, approaches to the trials themselves, outcome measures, no-go criteria, patient selection, ethics, inequality, and doing these approaches, as well as funding mechanisms, which are all sort of in the works. But some of the examples from clinic can still be very precision driven. So I have a child who I met in epilepsy genetics clinic who had not an SCN2A variant, but a variant in GRIN2A, a glutamate receptor, for example, and there are medications for other purposes, dextromethorphan, a cough suppressant, memantine, an Alzheimer's drug that actually target the pathway. And so we and several other centers developed a clinical protocol, but a protocol nonetheless, and treated those patients. And that little boy that I follow came to me having hundreds of seizures a day, severe spasticity, little to no interaction. And on those drugs, he's had a sustained more than 50% reduction in seizures, not complete cessation of seizures, but sustained, improved quality of life, and stayed out of the hospital. I have a couple of examples of kids that we've gotten INDs for medications that didn't qualify for trials, who went from monthly ICU admissions in with status epilepticus to at home with a much better quality of life. So there's real opportunity in these areas. And as far as how do you know whether your child is going to be eligible for one of these things, my general encouragement is to enroll in the research studies that are out there, the databases that are out there, so that variant is known, mechanism is known, and you can be contacted if something comes up that would be an opportunity that would be relevant for your family. Happy to answer other questions.
So if someone has questions, I'll walk the mic to you. Just need to see a show of hands.
So I know everyone's not really giving, and rightly so, but talking about specific drugs in particular or treatments for seizures. But I haven't heard anybody mention the vagus nerve stimulator. My daughter does have one, and that has helped immensely. So I'm wondering, what's your take? Do you see that as a treatment that's still questionable, or what are your thoughts on that?
Well, VNS is definitely an approved treatment for refractory epilepsy, not necessarily specific to SCN2A or the pathways, but an approved treatment for refractory epilepsy, and it has benefited some patients. I don't think I know of any SCN2A specific literature on VNS unless you do, but yes, it's definitely an option for children with refractory epilepsy, as is ketogenic diet, for example.
Heather, while you're still at the microphone, so the N of 1 trial that you just described sounds a bit uncontrolled and anecdotal. Can you convince me that it's not? I'm
So
being
the
controversial here.
examples I was giving,
Yeah.
is that what you mean? Yeah, so those were not necessarily formal N of 1 trials, but I was more trying to describe those as sort of the lead up to these more formalized approaches that we're going to need to be much more rigorous about when we're talking about something invasive, something disease modifying, but they're things that we've done in clinic. And yes, it's uncontrolled because there is no control. It's one patient. You could, and there's some debate in N of 1s of should there be a period off drug or a period of actual placebo. And those are things that can certainly be discussed. In these situations, it was more of a, as rigorous as we can be in clinic approach to creating baseline data, neuropsych, seizure counting, EEG data, following them for a period of time and having a follow-up and relevant to their natural history. And some of the N of 1 trial approach is really going to be very much that too. It's going to be making sure you really have a sense of that trajectory of natural history that's patient-specific, outcome measures that are very patient-specific and personalized, more so even than you can do in a larger trial where you're accounting for different phenotypes and setting ahead of time, what are your goals? What are you looking for? And what's going to be your no-go-go? I'm going to continue this drug or I'm not going to continue this drug. Does that answer the question?
Thank you.
It's a challenging area.
Are you going to read from the cards?
I got 2 from online, and it could be from our MNSAB or any scientist or clinician here that has some good content. So first question is, some gene therapies use stereotaxis with the aid of MRI to deliver drugs to the brain, since the blood-brain barrier prevents the action of most drugs applied to conventional routes. Would this be applicable in treatments for SCN2A? Anyone got it?
Everybody's looking. Yeah. Everybody's looking at me. I don't know that I have experience with this, but so you said stereotactic MRI guided delivery of the drug to the ventricles, to the brain?
Yeah, I think that's the.
I mean, they're using, in my experience, they're using the lower back to deliver some of these drugs and some of other trials that are ongoing. But if there was a particular reason to deliver it directly to the brain, that could be used. It's a very specific question. So maybe there's some planned study that I'm not aware of that might be using that technique. Do you know of one, Heather?
No, but I think it would really, and this is maybe a better question for anyone developing gene therapy out there. One of the speakers touched on gene therapy. Maybe she wants to comment. But I think it's really specific. There are different approaches to gene therapy to delivery in blood versus CSF and different regions to deliver. So I think it really depends on the details.
Yeah, I think one important thing to consider with this question is what areas in the brain is SCN2A important for? And I think we know some areas where it definitely is important. And I think there are still some questions on what areas of the brain a gene therapy would have to be delivered to. And in this approach, in both the intrathecal where it goes through the back or directly into the brain, you're just getting brain delivery. And if there is any SCN2A externally outside of the brain that's important, there wouldn't be a benefit there as well. All right.
Anybody in the audience want to comment?
I think certainly SCN2A is widespread in the brain. Outside of the brain, expression is much more limited. Whether or not you need it in some of the autonomic nervous system in these long nerves that are penetrating outside, that's a little bit less clear. But certainly once you go through the blood-brain barrier to do your delivery, you do have some leakage that comes out as well. So you might get sort of incidental exposure under those conditions.
Thanks. We got some questions for Kevin. First one, the CRISPR-A technology sounds promising, aside from the complicated issues of delivery. Can you speak to the durability or permanence of the effects of CRISPR-A and whether it is possible to develop a on-off switch for safety?
Yeah, I can. Hi. So CRISPR-A is an approach that utilizes viruses, and once those viruses get into cells, it has gotten into the cell and it's doing its job. The beauty and the advantage of the nervous system is it is both immune privileged, and once those neurons are born, they don't divide again. And so if you can get it into every cell, it should work for the rest of that neuron's lifetime, which hopefully is the rest of that person's lifetime. And then the other question is an on-off switch. Yes, I mean, the biggest questions in this field for Nadav, for Navneet, I should mention Navneet and Perry are part of a startup company that pulled off from UCSF called Regal Therapeutics that is advancing this approach, is to develop techniques to both miniaturize this as small as possible and also essentially make dimmer switches for this approach, as Will mentioned earlier. So it might be something where you would want to take this along with something, an oral pill that actually turns it on or turns up the gain or turns down the gain. Now that would have to happen right at the onset, because once it starts working, it starts working. And then, well, actually, no, you could turn it off afterwards. I think you could make a kill switch. You could design anything and those mice will be super happy. The translation is always the hard part.
We'll just keep you right there. We got one more for you. Other than delivery, are there other big problems to solve the CRISPR-A before it can move to a trial? Are there off-target effects? Is that a major concern?
Yeah, off-target effects are whenever you hear the word CRISPR, you think first of lettuce, and then you second think of what it's going to do genetically in terms of off-target effects. So CRISPR is designed to cut in its initial form, and these things called guide RNAs tell it where to cut. CRISPR-A is a particular case where we've removed the ability for CRISPR to do any cuts. And really what it is it's chaperone to a party. It guides something else to that part of the DNA and allows that other thing to up or down regulate that region. And so there is a concern. If you have something that guide could target in another region of the cell or another region of the DNA, it could have off-target up or down regulatory effects. And we've looked at that extremely carefully. Of course, SCN2A is right next to 1A and 8A. And you worry about having differences in effects of those genes. We've looked at those extremely carefully. That's actually published in a preprint on bioRxiv that you can download and look in the supplemental figures and look for changes in expression. Long and short of it, we haven't seen any changes. We need to do more, obviously. But so far, and I also say for Nadav, this is his third or fourth gene that he's tried. And in the first couple of genes, they've looked very, very carefully at off-target effects and have not seen anything. That's the beauty of this CRISPR approach. It can be very specific.
Thank you. We got two here for Catherine Myers. The first one is SCN2A too big for gene therapy? And are there other ways that you could make that work?
So the question was if there are other ways to make that work.
I guess the first is it too big for a gene therapy? And then
Yes.
if it is, then are there other approaches?
It's too big for AAV gene therapy as we know it at the moment. I'm sorry, I'm just trying to not have anyone in the back. Too big at the moment. There are other vector systems that are in development that might allow in the future moving forward with the whole gene or the whole protein at least to be transferred. But those are not as effective right now to get into the neurons. But there's still a lot of development around that. There are other ways to make it work. We can split up the protein into multiple pieces that when delivered, for example, would be stitched together. They come all with their own risks because you could end up in one cell with just one piece of the protein, and that could be toxic as well. So we don't have really good solutions yet. Of course, what Kevin presented or any modification thereof that doesn't require maybe a bacterial enzyme could be really interesting, because you could use the endogenous protein on the allele that is not mutated and just upregulated. Then you don't have to deliver the entire gene, right? So there's a couple of options that a lot of people are working on. There are non-viral deliveries that will allow to package it as well. So we just need to figure out the delivery and increase the efficacy of those approaches as well. So I think in the next 5-10 years, there's going to be a lot more options developed that could be used for gene therapy in any sort of way for these proteins.
You can keep the mic. We got 2 more questions for you. Do you see changes in cellular mitochondria energy states in SCN2A haploinsufficiency or only in gain of function mutations?
So that's the interesting part. We've actually seen it in both gain of function and loss of function cells, but we do need to increase the number of cells that we're looking at, which I'm really excited to get more samples so that we can see, is this a more general thing, or are there specific mutations in the gain or loss of function realm that could cause similar effects. We have one loss of function mutation that actually leads to an upregulation of the SCN2A protein. So there could be some sort of feedback loop that we can leverage as well. So I can't say how broadly applicable it is, but it might not just be a loss of function or gain of function observation.
And the last question for you is, you mentioned you're working on a drug that reduces the excitability of neurons. Can you talk any more about that?
No, right now we can't talk more about it because we are working with, different collaborators. So unfortunately, hopefully, I'm hoping next year or at the next meeting or anytime sooner than that, we will have more information.
Thank you.
Thank you.
Maybe this one's a good one for Dr. Olson. What would qualify an SCN2A patient to be in an N of 1 trial? Sorry.
That's a great question. So it would essentially be if there were a novel therapy that was developed that was specific to that individual. So something that targeted that specific variant or a SNP linked to their specific variant, for example. So if there were a precision antisense oligonucleotide linked to a single nucleotide variant that is, developed for that individual, or something that modifies, there's an example of an ASO that was called Melicin that's published, in the most widely known case out there that targeted a pseudogene, which is a unique mechanism unique to that child. And so she was treated with that in an N of 1 way. So it's really having a unique treatment approach for a unique variant. And that's why I say, I don't think everyone in the room should go say, what's my N of 1 trial? What's going to be my treatment? Nor is it necessarily the right answer for everyone. If there's something that is a broader, something that we can really prove efficacy in a larger trial, there are definitely advantages to that from a safety and efficacy standpoint. So it's not for everyone. And right now, I think the best approach is to make sure that your information is out there so that if there is a unique treatment approach, you'll be able to hear about it.
I think that's it.
Well, that concludes the Q&A with the Scientific Advisory Board, and thank you all for submitting questions. We are at the end of the program, and I want to give a thanks to everyone who participated.